Okay, so we can get started, and as I think I heard whispering, this is a question from the quiz, and overall everyone did do well. I think it was like a 74% or something like that average, and this was the sort of one question that was the sort of people struggled with the most. And so I thought I'd just kind of bring it back, even though I'm sure you all looked at like your notes right after the quiz to try and figure out how you were doing even before it was posted. So, but this is just to remind you that the way in which the long-term potentiation works, which is this long-term increase in synaptic strength, is that it's glutamatergic synapses, and the normal, most of the normal transmission is driven through AMPA receptors. However, if you have the postsynaptic neuron is active at the same time as the presynaptic neuron, that activity in the postsynaptic neuron will relieve a block on by magnesium on the NMDA receptor, which allows calcium to enter the postsynaptic cell, which is the initiating step for this increase in synaptic plasticity. And just, again, to remind you, there are some drugs that do act through NMDA receptors. So this isn't just for sort of, like airy-fairy neurobiology interest. It is actually sort of will have relevance, like once we get into the sort of pharmacology of certain drugs. The other thing I will say is that I was kind of hoping more for sort of like an 80-something%. And so I believe I'll be giving you another quiz like towards the end of my bit, and I'll probably try and figure out some way of doing a bonus question or something like that to try and help it get up to that level. They are both receptors. I guess I can get there with that one. So like if this is the postsynaptic cell or dendrite, I guess. So normally at normal resting potentials, you have when you have glutamate present, it binds to the AMPA receptors. Now you can read the AMPA, but that's it as AMPA. So glutamate binds to the AMPA receptor, opens the channel, and sodium comes in, and that creates an excitatory postsynaptic potential. Sorry, what is that? Could you write a little bit? So this says AMPA. Yes. And this says glutamate. This is why I don't sort of do all of my teaching with the whiteboard. So, however, if we have a situation where this already has lots of positive charge inside, like the membrane potential will be reduced, and under those situations, so normally the NMDA receptor is going to be closed, but if you have all of this positive charge inside, then magnesium will leave the NMDA receptor's channel, and that will allow sodium and calcium to pass through the channel, and that's what is the initiating step for the long-term synaptic plasticity. And there's a much prettier diagram of this actually in the slide set. So the mGluRs are the G protein coupled receptors, and they do have a modulatory role in synaptic plasticity, but we didn't talk about that because that gets kind of is more detailed than we need at this point. Okay, so now we'll go back to the slide set we were on yesterday, where we were talking about some of the higher functions and memory and so on. And we just finished talking about emotions. And the next step is talking about speech and language, because that's also a specialized function of like the cortices. So language is like fundamental to obviously human experience, because that's how is why you get to sit in a classroom listening to me talk. If we didn't have language, obviously I wouldn't be able to transfer that information to you. And you know, we don't have telepathy, so we have to convert the thoughts into, you know, like something that will transmit between people. And auditory language is, for most people, how we do it. Interestingly, sign language uses some of the same brain areas. So we're trying to sort of communicate to separate people, and we're trying to sort of interpret what they're saying. Obviously, if I were to, at least for most of you, if many of you at least, if I were to speak in French, then it would not be that easy for you to follow. If I was to speak in Russian, it would be a miracle because I speak no Russian whatsoever. So you know, like the mechanisms in the brain are not language dependent, but obviously we have to learn the language in order for the brain areas to function. So, and there's sort of multiple steps. So we have to think of the right words to use, and then we have to carry out the motor action that's involved in speaking, because, you know, like, it is obviously this very fine motor control of our, you know, voice box that is, at the end of the day, giving this, you know, shaping the sound vibrations that you're hearing. And so the we have two major areas that are responsible for speech. There's Broca's area, and there's Wernicke's area, and each of them is named after the person who discovered them. Basically from stroke patients, because Broca's area is involved in the sort of organization of all of the sort of motor activity that's involved in speech, and it also is involved in the ability to assemble words into a sort of meaningful sentence. And it's also located right next to the portion of the motor cortex that's actually responsible for, you know, triggering the areas of the lips and the tongue and so on. So obviously we want to have a bit of the brain that's responsible for generating speech, but we also need a bit of the brain that's responsible for understanding language. And that's Wernicke's area. And it's in part of the auditory association cortex. And there's a specific neural connection between these two areas called the arcuate fasciculus. And so you can have damage to that bundle, which will then separate the ability to create create language and interpret language. And that's called a conductive impairment of language. But damage to each of these individual things will cause very specific problems with language. Like, so for example, in Broca's area, the sort of very first published patient that he described was, I think it was called something like da, because that was the only sound he could make. And he could understand perfectly well, but he just couldn't, you know, produce any intelligible speech because he had, you know, like when he subsequently died, Broca, you know, did an autopsy on his brain and, you know, discovered there was this damage to this particular area, which, you know, like obviously afterwards was named after him. So there is some lateralization of speech as well, so that in most people, the speech centers are sort of located in the left hemisphere, while the right hemisphere, sort of analogous regions, are mainly processing emotional content of speech and understanding. And so there's this, we initiate our speech and in Wernicke's area, and then send that sort of speech planning to Broca's area in order to sort of say something. And then obviously Wernicke's area is also responsible when we're actually hearing someone speak, and, you know, in terms of generating an understanding. There are also, as you would probably expect, inputs to those areas from the visual cortex. And so that's how we're able to, for example, have visual language, which includes sign language, but it obviously also includes the ability to read, because that's also integral to language and uses many of the same, not the exact same pathways, but sort of also uses Wernicke's area for interpretation. And so patients who have problems with language are said to have aphasias. So you can have Wernicke's aphasia or Broca's aphasia or conductive aphasia. So they're all sort of like, but they're all of the aphasias are these disorders of language. So if we're kind of moving on a little bit, then we have the link between association and cognition. And this is this idea that we have every sort of thought that we have in our heads is essentially made of like different associations that we sort of build up of, you know, like little bits of, you know, like sensory information and little bits of accessory emotions all come together to form some kind of association in the hemispheres, cerebral hemispheres, that allows us to sort of experience, you know, like a thought. And so we have these, the different cortical regions specialize in different aspects of that association. But so for example, like the occipital lobe is, you know, like contains the visual cortex, but it also contains the sort of association areas which surround the visual cortex, which are involved in not just interpreting, you know, like creating the visual map, but actually involved in establishing the patterns of connections that form associations, which, you know, like allow you to sort of, you know, recognize a computer or, you know, like a person, things like that. And so we have specialized associations around particular sensory areas, and then we have more general association areas which are involved in more multidimensional thought. And especially the sort of frontal cortex is particularly involved in, you know, like connecting all the pieces together. For example, you know, like say, I don't know why I'm always using dogs as an example, but you have a dog, you know, like say you're petting your dog, you know, like there's a sensory feeling of what the fur is like. There's the sort of, you know, like maybe it makes a sort of happy kind of rumble sound when, you know, like you're scratching its ears. You have the visual appearance of the dog. You have the emotional connection. So all of those things come together in your sort of mental model of what your particular dog is. And so all of that final piecing together portion is sort of what's happening through the frontal cortex. And so that's, you know, you can see how there's all these different threads, different pieces of information that make up your, you know, like particular dog. And at some point in the brain, in order to have that concept, you have to sort of put them all together. And so this is why sort of association is, the associations between different brain regions are able to create the associations between different percepts in terms of, you know, like different pieces of sensory information in order to create an actual sort of thought about, you know, like anything. And so the same process is true for, you know, like anything else. It's nothing specific to dogs, but it's that multidimensional aspect that gives rise to, you know, like this is driven by the associations between these different cortical regions. And actually, this is basically what's summarized in this slide. So So what happens when some of that association goes wrong? That can lead to agnosias. And agnosias are disorders of recognition. So typically, because they're tied to a specific sort of, you know, like modality in some fashion, like whether it's, you know, like visual or auditory or something like that. But they can also be, you know, like spatial, as we'll talk about it in a moment. And the reason why they usually have stereotypic sort of like just portions of, you don't have complete agnosia, because to create a complete agnosia where you can't really perceive anything would essentially mean that you had damage to sort of really widespread areas of the brain, in which case you're probably not thinking at all. You're in a sort of deep coma, and then, like it's kind of a wash whether or not you could even sort of describe it as an agnosia or just, like complete brain failure. So usually they're going to have specific characteristics because they're due to damage to specific regions of the brain. And so, for example, you can have visual object agnosia. And in some cases that can be astonishingly specific. So like, sometimes, but in the general feature of visual object agnosia is that you cannot recognize something by looking at it. So if I was to stare at this, I'd have no idea what it was. But, and this is the thing that's kind of interesting about it, is that if I touch it, then I can recognize it. So, and like you could have, like also have a visual object agnosia where something is sound emitting, and the sound would be enough of a trigger to sort of generate the object recognition. So that's demonstrating 2 points. So one is that you can have these remarkably specific injuries to the brain's ability to recognize things, but that they can sort of, they are tied to specific modalities so that other modalities can reestablish that sort of recognition. And for when you're able to sort of look at this, obviously in terms of diagnosis, like if someone has advanced dementia, then you wouldn't really be able to tell whether or not they have like some kind of agnosia, or it's just part of the dementia. They may claim that there's, like if someone's in truly advanced dementia, then you're not able to sort of communicate effectively enough to be able to sort of distinguish that kind of feature. Another interesting aspect of, or example of agnosias are hemiagnosias. And they're sort of really kind of funky because there it's basically half of the world can't be recognized. And so we have these sort of cartoons, like on the right, or not cartoons, drawings on the right. So basically the patient is asked to, like just copy an image. And so if you look at this clock, you see that it's missing all the numbers on one side, but has them on the other side. The same is true of sort of trying to copy this picture of a house. It's sort of like half the house is missing. It's, there are people who have this, have trouble getting around because they sort of only see half of the street that they're sort of looking at. So there was an interesting example of where someone was asked to sort of visualize their sort of town square. I think it was somewhere in Germany. And they could sort of like accurately describe all of the sort of buildings on the right-hand side of the square, but there was nothing, as far as they were concerned, there was nothing on the left-hand side. Then they were asked to imagine themselves at the other side of the square. And now all of the buildings on the other side they could remember, but they couldn't remember anything on the left-hand side. So it's a complete inability to sort of experience one half of your world. But you can obviously turn around and then you get the other half, but you'd never get two halves at the same time. And it's to the point where, for example, these people are eating, they'll eat food from half of their plate, but not the other half of the plate. Yes. That is a very interesting question. So actually, no, they come up with these really elaborate stories as to why they're actually perfectly fine. So they actually are really hard to convince that they have this issue. And like you could show them that obviously these drawings, and they would say, yeah, no, it's complete. I drew like all the numbers all the way around. So I mean, I actually talk a little bit more about this I do a elective on sort of psychopharmacology, and we sort of talk a little bit about some of these things. But it's a perfect example of how much of our perception of reality is is driven by the particular functioning of bits of our brains. So it's really interesting. Yes? Yes, yes, exactly. Their two eyes are both completely fine, but they. So, for example, okay, this is on my left. My computer's on my left hand side. So like I could, so like this, like I can still kind of see it. It's there. If I had a left side agnosia, I wouldn't be able to see it through either eye. But I could stand over here, and then I would be able to see it, even though both eyes are able to look at it. Yes. They do not remember the object exists. Well, depends whether it's left or right side. But yes, if it's on the neglected side, it completely passes from their mind. Yeah. Yeah, your computer would wink in and out of existence. So I'm going to skip the talking just so we can kind of not get further behind, and so we can talk about get to talking about pain. And I will mention, so this slide will not have any questions from it because there aren't, other than sort of anesthetics and so on, there's sort of nothing that really manipulates consciousness at a sort of like, clinical level, but it's an interesting subject to talk about. And I should mention there are also, again, study questions at the end of this PowerPoint that you can sort of look at to try and, focus your study. Okay. So now we're moving from sort of deep thoughts about the meaning of existence and so on to something that no one has any doubts about, which is that if you smack your thumb with a hammer, it hurts. And so the question is, of course, why does it hurt? And how does it hurt, sort of in a neural sense? And so this class is then sort of leading to what will be Friday's class, when we'll actually talk about a particular type of pain, which will be headache, which will kind of be the beginning of going into sort of more the physiology underlying more actual disease states rather than sort of kind of an introduction to neuroscience. So as an introduction to pain, we're going to first talk about things that aren't painful, which is touch. So obviously I can rub I don't have anything wrong with my nervous system that I'm aware of. And so I can rub my fingers along this desk and it's not painful, but I can feel something. And that is obviously driven by touch receptors that are present in the skin. And there are multiple, as we'll talk about in a minute, there are multiple types of touch receptor in the skin. And they have an interesting property, which is that they all have their own receptive fields. And so this isn't something that this is where it's sort of like very different from other kinds of sensory information, where, it's like, this is my visual field. It's sort of like, I don't sort of have like little areas of focus within it. It's just like, one kind of very slightly blurry field because I don't wear glasses except when I'm driving. So touch information is varies in terms of its acuity, varies dramatically depending on what part of the skin it is. And even within the hand you see this. So each of these little circles is an indication of the sort of acuity of touch. And the way in which you test this is you literally have two little pins, and yes, and you sort of whether or not you can detect it as two pins prodding you or a single one. And so you go further and further apart, and then at some point you can distinguish this as two separate touches on your fingers. And so if you see right on the fingertip, you have very high acuity. And I've never quite understood why so many of these little blood things are sort of right on the most sensitive part of your fingers. But anyway, They should really be doing it sort of like somewhere like on the thigh or something like that, where you have very poor acuity. Yes, exactly. And this chart on the right, this the y-axis is the distance that you have to move apart before you can distinguish like two points prodding your skin.
So the higher the spike, the less sensitive the area is.
Exactly. And so what you'll see is that the hand, for example, has very high acuity. So does the sort of the lips and the tongue. Whereas, yeah, the sort of thigh is probably, and the sort of center of the chest, things like that, have the least sensory acuity. So what happens when we're touching, and sort of what gives rise to some of this different acuity? Well, it depends on which receptors we have in that particular area of the skin. And it depends on how much those receptors kind of are spreading out their like sensory axons. So the general process of sensing something in the skin is obviously there's going to be some kind of stimulus that's going to hit a mechanically sensitive receptor, which could be high threshold or low threshold. High threshold mechanical sensors are going to be the ones that make you go ow, because they're the pain sensors. What happens when you're touching them is that you get a change in the ionic permeability of the particular sensor, i.e. they depolarize in response to their, like being physically manhandled. So we get depolarization, we generate an action potential in those sensory axons, and then that propagates up to the CNS via the, so it goes to the spinal cord where there's a lot of processing, and then up to the brain itself. So, and we'll talk in a moment about these different types of sensors. The fine nerve endings, which are circled here, are the ones which are these high threshold mechanical receptors. And so those bare nerve endings are the ones that are going to be responsible for, like the sort of pricking pain if, like someone stabs you, or you get a, like a little blood test. So this is, breaking down just the touch ones. This doesn't include the pain sensors. So we have Meissner's corpuscle, Merkel cells, Paxinian corpuscle, and Ruffini endings. And these all have their different kinds of receptive fields. So we have the Meissner corpuscle and the Merkel cells have pretty tight receptive fields, whereas the Paxinian corpuscle and Ruffini endings are less sensitive. But in all cases, they sort of have this reaction to, like their sensation, which they encode as bursts of action potentials. And what you'll see for like most of them is that, so here's the example of the stimulus, is sort of what it means by this little diagram here is that there's sort of no physical stimulus, and then it ramps up in intensity, stays static, and then drops again. And so, for example, here we see the Meissner corpuscle is responding to increasing stimulus by a burst of action potentials, but when it just stays the same, then it stops firing. And then when that stimulus goes away, then it has a brief firing again. So it's encoding the type of, like physical interaction that it's experiencing. And that's a, fairly common type of firing pattern actually for many neurons, where they're sort of detecting changes rather than actual analog, like, or digital to analog to digital conversion of the signal. So we can actually, like this isn't sort of something that needs to be done in like animal models. We can do this with students, and we just give them like a kind of leather gag in their mouth in case they scream too much. But what you do is you have the obviously little probe to sort of prod their fingers, and you basically have an electrode in the elbow, and you can actually just sort of like prod the finger and detect the sort of sensory information as it's sort of flowing through the nervous system. And obviously this is non-painful because this is touch. And so what you can see is so this is an example of these recordings. And so like you give a weak little touch, you can detect a tiny little compound action potential in the nerve. If you give a stronger touch, so these are now, this is the sort of pressure that's being exerted by the little probe. So a stronger touch, you can see more action potentials firing, and even stronger, like you get more signal. And again, in both this example shows it more clearly. You can see the on signal and the off signal. So there's a burst for the initiation of the touch and a burst for the termination of the touch. And again, here we have a ramp for the pressure. And because it's ramping, there's a continuous changing of the pressure. So there's the burst when the sensation begins, and then like some ticking over because you're gradually increasing the pressure, and then another sort of little burst of action potentials to say the pressure's gone. So we're encoding these sensations into bursts of action potentials in these sensory nerves, which will then come up and sort of make contact within the spinal cord. So there's a number of different types of pain. And I'm sure you all aware of this from like personal experience. The sort of feeling of like accidentally cutting your finger feels very different from, say, the pain of a toothache. And if anyone's had appendicitis, that's like a different type of pain as well. So pricking pain is the sort of exactly what it sounds. It's like that very sharp pain when something, someone takes, I don't know why I keep talking about it, but someone takes, like one of those little blood tests from your fingertip. You feel it like immediately. Same as if you're sort of having an immunization or someone's taking blood or something like that. It's very fast, sharp pain. And it's very accurately localized. And typically, it will cause the sort of pain reflex, which is this withdrawal reflex, where you sort of feel something and then you instinctively kind of pull away. And it's not generated by any deeper tissues. It's specific to the skin. And it has the reason it's sort of the first pain is because it's transmitted very rapidly through myelinated A delta fibers, which have a speed of transmission of like 6 to 30 meters per second. So it's very fast, and it's very, it's accurately localized and very fast. The next kind of pain is burning pain. And this is typically more intense, but the localization of it is not so good. So if you like stab your finger, you can really visualize exactly where it is. If you sort of put your finger on like a hot electric stove or something like that, it's certainly painful, but you don't have that same precision with the localization of it. It's a slower onset pain. It's transmitted to the CNS via unmyelinated C fibers. And it's like some of the other more intense types of pains, it evokes a very strong sympathetic response. So with like heart rate, sweating, pupil dilation, and so on. Then we have sort of deep visceral pain that has a kind of aching, burning quality to it, but and obviously can be very intense. But and it's also mediated by these unmyelinated C fibers. And the visceral pain often has this characteristic that it's really very difficult to localize, and in some cases it's actually mislocalized because it presents as referred pain, which is what's illustrated in this diagram. So this is just an example where you can have, there are stereotypical, so referred pain means basically you're feeling the pain in a place other than where the actual insult is. And so, you know, like one of the classic examples is, for example, you know, like pain from a heart attack or something like that. You feel it's sort of this kind of shooting pain going down through you. Typically it's the left arm. You know, like most people don't have a heart in their left arm. So it's not in the sort of right place, but it has this stereotypical presentation. Similarly, kidney pain is typically this very broad diffuse pain that go, you know, like it's kind of lower back and then sort of down to the top of the legs. These sort of, the stereotypical nature of these does at least mean that you can get some diagnostic benefit from it. But it's obviously, you know, like required a lot of sort of medical information over time in order to sort of be able to sort of interpret these different types of referred pain.
I got a question. The kidney area, that seems like it's got a lot of overlap with normal symptoms of sciatica. How would you differentiate between whether the person is suffering from sciatica or whether it's referred pain from a kidney infection?
So, well, in many cases, sort of, it would depend on, well, you'd have blood tests, you would have, if you suspected the kidney thing, you would be doing ultrasounds and things like that. So in many cases for neurologic types of pain, you eliminate all of the other options, and then you sort of say, yes, yes, exactly. So I'm going to push on through, just so we can try not to fall too far behind. But so it's the sort of, I'm going to just give some key takeaways. So we're going to, so we have our different types of sensing in the skin, right? So, and their response, so we have these different sensors in the skin, and we have low threshold mechanoreceptors and high threshold ones. And so what do the high threshold receptors do? Yes, exactly. And which are, if we have different types of pain, and so what type of pain is fastest? Yes. And then, and what kind of axons transmits visceral pain? Yes, perfect. And whereas the pinprick pain is transmitted via myelinated axons. So it all kind of fits together with what we were talking about excitability and action potential propagation speed. Yes. So it's, for referred pain, it's not quite the case. Well, so the explanation is that there's the actual sensing of the pain, like in the heart or whatever, it's not, it doesn't go all the way up to the brain, but so it has its own axon sort of transmitting that pain to the spinal cord, but within the spinal cord, then yes, it kind of gets lumped in with other areas and that gets sent up to the brain. So not at the level of the periphery, but once you get to the spinal cord, yes. What's that, sorry? Oh, the receptive areas. So most of the separation with the, so there is, for some of the very large areas, it is due to that kind of crossover in the spinal cord. But for the smaller areas, it's actually the receptive areas of the neurons themselves. So okay, so talking specifically about pain, we have to talk about nociceptors. And nociceptors is basically the fancy word for saying pain receptors. And nociception is obviously detection of pain, or experience of pain, rather. And so we have mechanical ones, which is, you know, like pinprick pain and so on. But then, and then we have thermal nociceptors, which are, you know, like where you're detecting sort of intense heat or intense cold. And then we have polymodal nociceptors, which are generally detecting chemicals. And so we have, for nociception, we have A beta and A delta fibers, which are fast. And then we have our C fibers from some aspects of thermal and polymodal detection, which are going to be slower. And how do they work? How do they sort of detect their different types of pain? So we talked about the free nerve endings responsible for pricking pain. Exactly how they work is at this point not very well understood. The thermal sensors and polymodal sensors are actually rather better understood. And thermal sensation, for example, can be, like it's usually classed as nociception, but obviously there are pleasant temperatures as well. Like, like if you have, like a warm bath, you don't jump in it and scream. Whereas if it was like, say, boiling point, you might well scream. So there's a difference. You can detect different temperatures, and it's only above and below a certain range that they become noxious temperatures. So whereas if they're sort of roundabout body temperature, they're innocuous temperatures. And then these polymodal ones, so temperature is detected through these receptors called TRP receptors, TRP channels, and the polymodal ones have specific ability to detect different types of chemicals. So whether it's they're detecting protons or ATP or histamine, things like that. So the thermoreceptors, as I mentioned, use these TRP channels. And there is a kind of nice little tie-in to taste, which is that if you were to think of, say, a taste that you thought was hot, what would you think of?
Birthday chilies.
Yes, any kind of chili. And so it's obviously the capsaicin in chilies that is responsible for the, like spiciness. So interestingly, the receptors that detect high physical heat are actually also the ones that detect capsaicin. And these are these TRPV1 and TRPV2 channels. So they're responsible for detecting, like painfully high temperatures, but they're also why your chili feels hot. And if you think of what's a taste which would seem cool? Yes, so menthol, and the TRPM8 channel is responsible for innocuous cool, and is actually sensitive to menthol. Yes.
So the chili activates the hot receptor, right?
Yes.
So how come you got the chili in my hand? I got like a burn.
A physical burn?
Yeah.
Because capsaicin is a physical irritant. That's how it's
yes. I mean, you'd need a super hot chili to, I mean,
that's the voice of somebody who's never cut jalapenos and touched their eyes.
Well, no, on the eye, absolutely, it does hurt. We have habaneros quite often at home, but yes, I mean, it's definitely doing that. And my wife wears contact lenses, and so sometimes we are organized in how I actually cut the chilies, but often not. So, what's that? Yes, in very high, yes. And so this one for noxious cold is, it is, or horseradish. And that is also something that we experience as hot, like taste-wise, but is actually, if you imagine, like if you put your hand in ice water, for example, it almost does feel hot when you go into it. It's because it's painful. And so that pain is mediated by the TRPA1 channel, which is the one, yes, that's responsible for detect the taste of wasabi. So actually, that's more of a pharmaceutics question, because these are lipophilic compounds. And so this is why, for example, Indian cuisine uses a lot of yogurt and so on, because that is obviously full of lipids, and so that can kind of draw it out. Whereas water itself, these things aren't very soluble in water. And so they sort of, once they're there, they stick around. So we have this, the point of this is that we do have these different channels which are specialized for detection of different temperatures. And they have the, these lines are the sort of kind of arbitrary, like the way they're drawn, but they sort of give an indication of the way in which they respond to increasing temperature. And we have multiple of them because we want to be able to cover a wide range of different temperatures. We want to be able to tell if something is, like warm or cool, but we don't want to experience pain when we're doing it, because obviously, like day-to-day variations in temperature, happen all the time. We don't want to sort of be in pain at midday every day and in pain at sort of 5 in the morning because it's got warm and it's got cold. But we do want to be able to sort of sense when things are getting sufficiently warm or cold, hot or cold, that they have a possibility of damaging our skin. What about other things? So we have, this is the cold receptor we've talked about, and the hot receptor. Then we have specific receptors that respond to protons. And we have ones that respond to ATP. And would anyone like to sort of speculate as to why it would be useful to have a receptor responding to ATP for pain? What that? So where would we find ATP usually? Or any cell, right? But like inside or outside of the cells?
Oh, inside, so it detects cellular damage.
Exactly. Exactly. So that's the benefit of sort of detecting, using ATP as a signal for cellular damage. And so if our cells are sort of like being broken apart, then something isn't right. And so we want to be able to experience that as pain. Because at the end of the day, that's the whole point of pain, is to sort of say, uh-oh, and I want to try and find some way of avoiding any further pain. It's these nociceptors are interesting from a sort of cell biology neural point of view, because they're unusual, where they have axons that are both receptive and secretory. So, like we talked about the sort of quintessential neurons with their axons being the output and the dendrites being the input. Sensory neurons are a little bit different because they're kind of a combined axon dendrite type thing. And so, and that has implications for what happens in the skin when it's experiencing pain. Because they have these sort of big branching neurons that cover these sort of relatively large receptive areas. And so we have a situation like in this diagram, for example, where we have a tissue injury. We've like cut into the skin. We've created obviously some sharp mechanical pain from the high threshold mechanoreceptors. And we've also released ATP and so on, because we've damaged the skin. And we that sort of activates this sort of sensory receptive axon, which will then branch to different areas. And these will, for example, activate mast cells and neutrophils to generate an inflammatory reaction. So we'll start getting things like histamine being released. We will also activate branches that connect to blood vessels. And so we'll end up with a big hodgepodge of different messengers all being released to the site of the injury, as well as just sort of the byproducts of cellular injury. And so this combination of different secretion of responses to injury, together with sort of the actual injury itself, creates this inflammatory response at the site of the injury. And this is basically promoting local sensitization, so that if, for example, you repeatedly injure the same part, it gets more and more painful. I don't know if anyone's ever had one of those sort of allergy skin tests where they sort of like stab you with a needle like about 50 times along your arm. And it's like the first 10 times don't hurt really, but like after a while it really starts getting painful. Just because you're triggering more and more sensitization. In the case of an actual, yes.
So let's say you like take some illicit drug, and make my receptor more sensitive to the drug. Maybe it makes more feeling pleasure or more. Maybe it makes sensitive to more pain. Would that make me more addicted to the medication?
So the sensitization in addiction is a different mechanism. So it's I don't know of drugs that make you more sensitive. I think there are drugs I can't think off the top of my head of drugs that make you more sensitive to pain, but most addictive drugs make you less sensitive to pain. But so these receptive secreting axons are responsible for this inflammatory response, where they're generating vasodilation, increased vascular permeability, and increased pain to the site of the injury. And so we have this hodgepodge of different chemicals, and so they're creating this multi-combinatorial effect. And the point of this is obviously to help with healing, because you're promoting blood flow in order to help with healing. You're also promoting, the reason for the wind-up increase in the pain is obviously to persuade you to sort of be a little bit more careful about whatever it is that got hurt. And this has a direct, you can sort of visualize this with what's called the triple response. So it's kind of shown up here, like you have a little cut. There's an initially it's just a cut, and then you create this little flare around it, and then the final stage is this wheal. And it's due to the release of histamine, from which we showed in this diagram up here, because through the activation of mast cells. And so first we have the sort of red spot, which is capillary dilation. Then we have the flare, which is the dilatation of these arterioles due to this sort of axon reflex. And then we have this raised wheal, which is due to the exudation of fluid from capillaries and damaged venules. And one of the interesting things is that this reflex, that is just a skin reflex, is actually often lost in patients with certain types of peripheral neuropathy. So for example, people with advanced diabetes, they have peripheral neuropathy, and actually these skin reflexes that seem like they're sort of entirely, would have nothing to do with the nervous system, actually disappear. which is like one of the signs of peripheral neuropathy. And so this is where we will stop for today, and we will get back to finish this on Friday, and then we'll also talk about headache. Thank you.